The core symptoms of autism are problems with language and language development, problems with social communication, social interactions, and rigid repetitive traits. We have no medications that treat the core symptoms of autism. We knew from other studies that serotonin levels in the bloodstream were elevated in a significant number of individuals with autism, about 25 to 30%. Key behavior that we now know serotonin controls is social behavior. And this, of course, is one of the changes that we see in autism. One of the major proteins that we discovered and cloned back in 1991 is the serotonin transporter. And many people know about this protein because it's a target of the antidepressant drug class typified by Prozac. We began to take apart the serotonin transporter gene in children with autism, and we found 5 rare but very interesting mutations. These mutations changed the function of the serotonin transporter in a surprising way. They didn't disable the protein. They actually made the protein work better. Normally, your serotonin transporter's activity needs to go up and down as serotonin is used. These mutations cause the serotonin transporter to go into high gear and stay there. We took the most common of these mutations, found in about 1% of the US population, and introduced it into the genome of a mouse. We call that mouse the CERT ALA56 mouse, named after where the mutation lies. When we analyzed the mice, what we found were changes in their behavior that were reminiscent of what you see in individuals with autism. We found repetitive traits. We found problems with communication between juveniles and the mother. We also found changes in social interaction. Perhaps even more interesting was that when we looked in the blood, we found the elevated serotonin levels that had been seen as a biomarker in humans with autism.
These autism mutants were sweeping away serotonin in a different manner than their counterparts. We also found these similar effects were present in ALA56 mice in our knock-in mouse model of these autism mutants. And so that left us really questioning, what are the signaling pathways that are regulating serotonin in the brain, and how are these things altered by these mutations?
We went looking for a drug that could target the enzyme and block its ability to regulate the transporter. Luckily, our colleagues at Northwestern had recently developed an experimental drug that did just that. And moreover, it gets into the brain easily. The next step was to give the drug to our mice in hopes of reversing the traits they exhibited.
We gave this experimental drug once. It had no effect. When we gave the experimental drug treatment for a week, we found no differences between our mutant mice and our normal mice. To be more certain of the results, we genetically altered the signaling with this enzyme and found similar results to our drug. One of the important aspects of our work is that the studies were conducted with adult animals. One of the things we need in the autism field are treatments that work in adults as well as children.
There are many years between this discovery and the use of a drug like this in humans. We're excited about the potential of using this drug or its sister drugs because the early studies in animals show it to be safe and actually effective in a number of disorders that involve changes in the brain.